All right, everyone. I'd like to welcome you all. We'll get started here. All right. Thank you all for showing up. I really appreciate when we do these things. So many of you come out. So this is really cool. Yeah, I guess this is our second lecture of the year. I hope to be giving one more lecture for an exam review. I was hoping to partner with the Biomedical Students Society for this lecture. Unfortunately, that didn't work out, but hopefully we'll be getting that happening for the final lecture. Maybe there'll be some food, something like that. We'll see what happens there. I guess I should introduce myself. I'm Jordan. You guys probably already know that. But the key thing you need to take away here is that I'm not a TA. All right? I'm writing this test just like you. So I have no special insights about what's going to be on the test. I'm really just trying to present the content that we've covered in class in a fresh way, and hopefully that you can understand it better. That's my Instagram right there. If you're not already following it, you can. It's not a personal Instagram. It's specifically for the lecturing that I do. And I've also recently been elected to the Senate thanks to votes from many of you. So thank you for that. Yeah, so if you want to follow what I'm doing, that's right there. Okay, I have one more thing to get to before we get to the content here, and I'm going to briefly go over this now, and then if you'd like to become involved with it, you can, we'll talk about it at the break and at the end of the lecture. So you may know that WUSA is our undergraduate student association here. WUSA is hosting its annual general meeting on the day of our test, Wednesday, March 27th, 5 to 7 p.m. And it's in the SLC Great Hall. Okay, I know most of you are not going to attend this, and that's okay. I probably wouldn't either if I wasn't interested in politics or the Science Center, but I'm going, and maybe some of you will too. So what I want to propose is that we create a science super vote, as the name suggests, okay? So here's the basic idea. That's all of us right there. That's us in the audience, okay? What we can do is take a small group of science students, the students who are going to go to that general meeting. So I'm thinking 4 or 5 of us. If you are planning on going, please stay after so we can work this out, okay? What we do is we take all the votes from that group, and what you can do is you can proxy your vote. That is, give your vote, if you're not going to use it by attending, you can give your vote to another person. So what I suggest we do is we take all of our votes in this room, pool them together, give them to this group of science students here, who then decide how they're going to vote. We come to an agreement in that group, and then we vote at the meeting, and basically we could outvote anyone there. And you may be asking, Jordan, is this legal? Absolutely it is. I went and talked to the WUSA people, and they're like, never seen this done before, but you could do it. So that's the goal here. Basically that some science students show up to this general meeting and determine the policy of WUSA for the next year. That's basically what we're trying to do. So we're going to work this out. I'll show you how you can proxy your vote later on, because I know you guys are here for biology and not for politics. But man, that could be really cool. I mean, look at the size of this vote, man. Like, how crazy is that, right? Okay, so keep that in mind. All right, so the first topic that we're going to discuss today is linkage. That's the first slide deck in the content, so hence I'm talking about it first. Now, this is a pretty complicated subject, so if you haven't got it yet, have some grace for yourself. You might not get it right away, but try to stick with it, and it's going to take a little bit of effort on my part to communicate this well, so have some patience for me too. Okay, so the first question that we consider when it comes to linkage is about Mendelian independent assortment. Mendel's laws tell us that different genes inherit independently. That is, the inheritance of one gene has nothing to do with the inheritance of another gene. Like, you can inherit 2 recessive alleles over here on the eye color gene, and then that in no way affects, say, your hair color, right? That's the underlying premise. But after Mendel, we found out that it's not the genes themselves that assort independently. It's the chromosomes that assort independently. Those are the units of inheritance that Mendel was talking about long ago. And so it kind of begs the question, what happens when 2 genes are on the same chromosome? Like, what goes on there, right? Are they inherited together? Does that happen all the time? What's the frequency of that? That's what we're discussing when we talk about linkage. So what I want to do here is help you visualize linkage when it comes to the actual chromosome and give you a definition. So linkage is when genes are close together on the same chromosome and they're inherited, resulting in the more frequent passing on or inheritance of the parental combination of alleles. The closer that two genes, or you can call them loci or a singular locus, that is where the gene is on the chromosome, the closer two loci are, the more linked the genes are, and the more frequent the parental combinations of alleles will be in the offspring, okay? The other thing to note is that genes on the same chromosome can, in fact, not be linked. If they're really far apart on the same chromosome, they won't be linked at all. Okay, so I just want to direct you towards these images here. On the left, you can see that we have Mendelian independent assortment, okay? Now, I've shaded the whole chromosome a certain color, but what I want you to envision is that there's a certain spot on that chromosome that has the gene that we're talking about on it. So you can see that on the far left, we have a set of two chromosomes that have one gene. Say that's the eye color gene, right? And then on the set right next to it, we have, say, the hair color gene, right? So eye color and hair color. They're on separate sets of chromosomes. When we're talking about linkage, those two genes are on the same chromosome. So maybe the eye color one is down here and the hair color one's up here, whichever it is, right? There's two different loci. And then, of course, the two different alleles are on different chromosomes, right? One allele on one chromosome, one on the other, but the genes are on the same chromosome. That's what I'm trying to get across here. So hopefully you can envision that. Two genes on the same chromosome. Okay, so this is probably going to be the most complicated slide for today, and this is where I introduce to you an experiment that determines linkage, okay? So what we're trying to do here is observe linkage in the offspring. What we do whenever we're trying to determine linkage is we start with a parental generation that is true breeding. Why would we do that? Well, we're trying to produce an F1 generation that is a dihybrid, okay? So it's heterozygous for both genes that we're looking at. Now, contextually, when we look at this slide, what we're doing is we're talking about the X chromosome. We're not talking about an autosomal one, the X chromosome. So the Y chromosome comes into play with this. The two specific genes that we're talking about here are body color and eye color. So you can see the parental generation there. We have a pure breeding female and a pure breeding male. The pure breeding female is white-eyed and has brown body. Okay, the white eye is the mutant trait, and the brown body is the wild type trait. And then the male has red eyes, so that's the wild type trait, and a yellow body. That is a mutant trait, okay? So then when we cross those pure breeding parental organisms, well, they're Drosophila, right? We get an F1 generation that has only one type of female and only one type of male, okay? And this is the way true breeding parents work, right? You only get one type of offspring. Because we're looking at the X chromosome, we have to separate this into males and females. Hemizygous, and then the females having two copies of each allele. Okay, so the key thing here is that we've produced a dihybrid offspring. An offspring that is heterozygous for both genes. Okay, so that's what we have in the female. Notice the male is not. The male only has one copy of each allele. It's the female that is truly dihybrid. So now what we want to do is we want to test cross the dihybrid F1 generation, specifically the female. Now, when we're looking at an X chromosome, instead of taking a male that is, recessive at both genes, like it's homozygous recessive for both, instead we can just cross it with a male from the F1. Why can we do that? If we only look at the males in the F2, we can ignore the contribution of the male. Remember, whenever a male is made, they have an XY chromosome. Where do they always get the Y from? From their father, right? So we can forget the paternal contribution to the F2 males. We're only looking at the maternal contribution to the F2 males. And then we can look at the way that the alleles assort in those F2 males. Now, all along, we've had sets of parental alleles, okay? So if you look at the parental generation again, right, and you can see that there's these slashes, right? Those indicate which alleles are on which chromosome. So you can see that for the female in the parental generation, one of the alleles is wild type and one is mutant on each chromosome, right? And you can see the separation there. So you can see the parental type from the female. It's that we have a mutant W there and a wild type body at the Y. And then it's the opposite for the parental contribution of the male, right? There's another parental type, parental combination of alleles. So now what we do is we look at the F2 males and we try to look for those parental types, right? And what we see is that the overwhelming majority of F2 males have the parental combination of alleles in red and blue. You can see them there, right? And then a vast minority have the recombinant types. That is pairs of alleles that are not what we observe in the parental generation. So the explanation for all of this is that there are two genes on the same chromosome and that they travel together. So the chromosome has traveled from the parental generation down to the F1 generation, down to the F2 males. And those combinations of alleles that were on that chromosome just stay there. And very rarely do we get recombinant types. That is times when the alleles are switched. They're switched from what they are in the parental combinations. So we've been talking about recombinant types. I want to help you visualize, instead of looking at the organisms, I want you to look at the actual chromosomes. I want you to see what a recombinant type looks like and how this inheritance pattern, the one I just described, looks like on the chromosomes. So if you look at the far left, you'll see a cross series A, and this is exactly what we just talked about on the previous slide. So we have the parental generation, again, pure breeding, and you can see that on a singular chromosome, on any given chromosome there, you can see that there's one wild type and one mutant. That's the combination we have. So there are parental types. We create an F1, and we get a dihybrid female. You can see that on the left there. We cross it with a male and look at the F2 males. That way we can ignore the contribution of the males from the F1 generation and just see how the inheritance of the X chromosome occurs between the F1 and the F2. So you can see there, right, the dark green and the light green chromosomes are preserved. They stay the same color, okay? You can see how they move down through the generations. Only rarely do they ever become recombinant. Are they ever half light and half dark, right? And the other side here, this is the same situation, except what we're doing is we're changing the alleles that are on the parental chromosomes. So we still have pure breeding parents, but instead what we're doing is we're making the female have all wild type. All of the alleles are wild type. And then the male, he has all mutant types. So you see that, right? The parental types are different. The same principles apply though. You cross them and you get a dihybrid female. The difference now is that her dark green chromosome has two wild types and her light green has two mutants. If you look over here though, the dark green had one mutant and one wild type. Right? So we're just changing what the parental types are, but the principles still apply. So you go down again, you look at the female contribution from the F1 in the F2 males, and you'll get, again, a 99% occurrence of the parental types. Noticing again that the parental types are different. So the assignment of parental doesn't apply to, arbitrarily like one wild type allele and one mutant allele. It's simply what those parental types are in any given experiment, okay? So just look at that parental generation, see the combinations of alleles. Those are the ones that you're looking for in these F2 generations. So recombination, we need to get into this a little bit more. What actually is recombination? Well, it's crossing over. You'll remember this from our previous test, right? Synapsis occurs, and then there's transfer of fragments of chromatids, the non-sister chromatids, and so what you get is a genetic recombination. And this is what we're investigating here. And really the point of all this is to show you that just because crossing over occurs, it doesn't mean that there will be a recombinant type for the specific loci or genes that we're looking at, right? What has to happen is there has to be a cut between the two loci that we're looking at. So you can see on the left side there, crossing over occurs, right? We have a combination of light green and dark green, right? The difference here is that the cut doesn't happen between A and B. On the right side, the cut does happen between A and B. So you can see that right there and right there. So when the cut happens there, that's when we have recombination within the context of the two genes that we have, A and B, right? Say, for example, we were looking at two different genes. How about C and D, right? And C and D could be positioned such that the cut on that first set occurs between them, and then we have recombination there, but there wouldn't be any recombination on this one because that cut occurs somewhere else. So that's the mechanism of recombination, crossing over at synapsis in meiosis. Now we talked about the example of the X chromosome, right? We also need to talk about the autosomal cases, all of the other chromosomes. What happens there? How do we set up an experiment to determine if linkage is happening in those chromosomes? So what we do is we follow the same procedure, we just can't rely on there being a Y chromosome. So let me walk you through this. We're gonna have our parental pure breeding types again. You can see that up at the top. They're separated by slashes to indicate which alleles are on which chromosomes. Notice though, that you're gonna have the same type for each gene, right? There's gonna be, so if we look at the far right at the top, there's gonna be 2 wild type for the B gene and 2 mutant for the C gene. Okay, so they are, I just remember this is C gene, yeah, okay, anyway. Yeah, so you can see that they are pure breeding, but in fact, they're just on different genes. Okay, so then we create an F1, right? And that is dihybrid. That is dihybrid. And you can see the parental types on the dihybrid. Then, instead of relying on the male having a Y chromosome and just ignoring the contribution of the male, we have to do a test cross. Okay, so we have to cross whatever this F1 is. So we might just take the females, for example, and then cross them with males that are recessive for both alleles, right? We could also switch this around. We could take the males from the F1 and then test cross them with a bunch of females that are homozygous recessive for both genes. It doesn't really matter whether we take male or female because we're looking at autosomals, right? It really matters though that we do a test cross because we want to isolate the contribution, isolate the chance from that F1 dihybrid. So then we can just take a look at the progeny, right? And we can see which ones are the parental types and which ones are recombinant types. Again, parental types being the combinations of alleles that you see up in the parental generation. And in this case, it's not as drastic as the last one. We have 77% parental classes and 23% recombinant classes. So we can say that these genes are a little bit less linked, but they're still definitely linked. All right. Now what I want to do is ask you a question. This is going to take a little bit of thinking, so if you got a piece of paper or something, you might want to do that. You could also logic it out in your brain, so give it a go. But here's the question. If there is 100% linkage, so remember we were looking at cases where there are sometimes recombinant types, right? There's 100% linkage in an autosomal dihybrid cross. What phenotypes will be expressed when the parental generation of these two pure breeding parents are crossed and the F1 female is test crossed, just like what we looked at before, with a homozygous recessive male? What types are we going to see in the offspring? Give that a think. So remember the process that we talked about, right? We have pure breeding parents. They create a dihybrid generation. You arbitrarily choose the female, test cross it. What offspring are we going to see in the F2 if there is 100% linkage? Do we have some ideas? Does anyone want to share their answer? Yes?
So for the F1 generation, would the options be like first is like half uppercase, half lowercase with the uppercase first, and the second option would be like half lowercase with the lowercase first? So would it be like capital A, capital B / small a, small b?
Yeah, that's right. That's right. It doesn't really matter which chromosome, like whether you put the capital A, capital B first or last, just so long as you've got the two parental types in there, right? Okay. So what would the F2 look like?
The F2 could have a recombination of the homozygous traits, so like you could have, so I'm assuming it would have like a 1:2 ratio of like the heterozygotes to homozygotes.
So remember, there's no recombination because we have 100% linkage.
Oh.
Imagine that, like 100% linkage. So we have no recombinant classes. Basically what I'm getting you to do is identify the parental classes that we would see in the offspring and their phenotypes that would be manifest. Any thoughts about what that would be? Yes?
So would they fully resemble the parental generation?
Would they fully resemble the parental generation? I guess they would. Yes, they would. That's right. Yes. But the genotypes would be different. So let's just take a look at the solution here. If you take a parental cross, right, of all capital A's and B's versus all lowercase a's and b's, you're going to get an F1, right, one contribution from each parent, right, of capital A, capital B, and lowercase b and lowercase a, okay? You're going to get that. Does that make sense? We've got a dihybrid with the parental type from each parent, okay? Then we test cross that. A test cross means that you're homozygous recessive for both alleles. So the contribution from the test cross will always be lowercase a, lowercase b. It's always going to be that. The question just is, what are you going to get from the F1 female that we're talking about here, right? And if it's 100% linked, that is, there's no recombination happening, you're only going to get the parental types. And the parental types are capital A, capital B, or lowercase a, lowercase b. So the two genotypes you're going to get are right there. You can see that. And then phenotypically, right, this is just like the parents again. One half is the dominant phenotype, and then the other half is the recessive phenotype. Does that make sense? I know this is a lot to digest. It's kind of hard for me to explain as well. But give it a think. If you need to read the textbook, do that too. But that's what we're talking about when we discuss linkage. We're going to keep the conversation going, but we're going to do that within the context of mapping a chromosome. We want to figure out where the genes are exactly on the chromosome and how they relate to each other. So what's the order of genes on that chromosome? And we can do that by looking at linkage. So first off, we need to discuss the term recombination frequency, or the RF value. Okay, this is really, really easy to calculate. If you can just see there, the formula is right here. The number of recombinants divided by the total offspring times 100%. Okay, very simple to calculate. In fact, this was calculated on previous slides, right? You could see that 77% were the parental type and 23% were the recombinant types. So that's the recombination frequency, the RF value. Okay. Now, what we can do is we can take the amount of recombinants that occur and turn that into a physical measurement. So if you think about it, right? If I have two genes, the farther apart they are, the greater the likelihood that a recombination event is going to occur, or a crossing over is going to occur between them, right? The farther apart, the greater chance there is that a cut comes in there and separates them, and then we get a recombinant type. So the idea is that somehow we can take the amount of recombinations that occur and kind of make that represent the distance. So we just do a really, really simple ratio here. We just say that the recombination frequency, 1% of that is 1 map unit. So for every 1% of the time that there's recombination, there's 1 map unit separating those 2 genes, okay? So if I have 23% recombination, there's 23 map units between the 2 loci of the genes. Okay, that's what we're saying here. Now what we can use are 2-point crosses. So 2-point crosses are what we were just doing before with the X chromosome and with the autosomal chromosomes there, where we were looking at 2 genes and trying to figure out the percent of time, or the percent of offspring that are recombinant or parental, okay? So 2-point crosses can be used to roughly estimate the distance using this conversion, right, from recombination frequency, RF value, to map units, okay? And what you can do is establish distances between loci. So from calculations like we just did on previous slides, you would get the distance between Y and W to be 1.1 map units, or 1.1% of the time there is recombination between the Y and the W genes. Then you'd get a map unit distance of 33.8 between W and M, and then 34.3 between Y and M. And you gotta remember, this has to be self-consistent, okay? If you get these measurements, what you can't do, for example, is say that M is on the far side of Y. Why doesn't that work? Well, the distance between Y and W would be fine, 1.1, and the distance between Y and M would be fine, 34.3. But if you think about it, if I put M all the way over here, right, the distance between M and W would be massive. It would be the distance between M and Y plus the distance between Y and W. And we calculated that the distance between W and M must be less than the distance between Y and M. So it's basically this logic. If you just sit there and think for a while, you can arrange them in the correct order. Okay, so it's a self-consistent linear order of genes. Now, this is quite an obvious question, but I just want you to think about it so that you're aware, okay? Why are the loci mapped, or the genes mapped, in a linear fashion, so a self-consistent linear graph, versus, say, a circular one or some other shape, right? Why is it that we only look at a line? Any thoughts? Yes?
It's easier to visualize like the distance in a straight line versus going around a bend.
Okay, the problem with the circle, I'll kind of give you a hint, is that it's like connected on both ends, right? What is it about like a circle, for example, that doesn't correspond to reality, right? What are we trying to make a map of? Yes?
Like you can get YM.
Yeah, like logically that's a huge problem. Yeah. What is it about the physiology? Yes?
Eukaryotic chromosomes are linear.
They're linear.
Like prokaryotes are circular.
It's a very obvious answer, but it's kind of hard to catch, okay? The idea is that eukaryotic chromosomes are lines. So we map out the positions of the genes in a line. If we were looking at, say, a prokaryote, we would have to do this in a circle because there would be a circular chromosome. That's what we see in bacteria, for example. All right, so that's why we're mapping this linearly. That's basically what I say there, so we can just keep going on. Now, you may have noticed this in the diagram that I just put up there of that 2-point cross example, but there was a problem with it. There was some math that didn't add up. What's the problem with using a 2-point cross and creating a diagram with that? Someone remember? Yeah?
Like map units aren't 100% accurate, so like if between like Y and W and W and M, the adders together, it's not equal to the distance between Y and M.
That's right. If you were to take, like, let's go back here just so you can see this. There. 1.1 plus 33.8 does not equal 34.3. What's up with that, right? Why is it that when we calculate the distances between 2 genes using a 2-point cross, we don't get a consistent mathematical answer? And that's the answer slide, basically saying what you just said. But you can see this on a greater scale, right? If you sum together, that's down at D, sum together all of the distances between subsequent loci, you're going to get 55. But if you just straight calculate the distance between Y and R, the outside ones, you're going to get 42.9. That's a big discrepancy, right? So what's causing that discrepancy? Well, let's take a look. The problem is we're not considering double crossovers, okay? Now I'm going to get to a slide very soon that's going to talk about the double crossovers, but what's going to help us increase the accuracy of our measurements is using a 3-point cross, okay? So instead of assessing just 2 genes at the same time, we assess 3 genes at the same time. And we can use those offspring to try to figure out a better approximation of what those distances are. So the fundamental difference here is that our experiment is not to produce a dihybrid F1, we're producing a trihybrid F1, okay? So we still have the parental generation that's pure breeding, right? That's still the same. And we're still producing only one type of offspring from that pure breeding cross, and that F1 offspring is a trihybrid, okay? You can see the parental types there, all mutant or all wild type. Then if we do a test cross, as usual, right? A test cross with that F1, doesn't matter if it's male or female, in this case, again, we're looking at female, we get these offspring, all right? Now, we can't interpret this right away as saying some of these are just straight, like, recombinants of all of them. We have to take a look relative to 2 genes, okay? Although there are 3 genes that we're looking at, in order to come up with a recombination frequency, we have to pick the 2 genes that we're going to look at. So in this case, we're going to take a look at, what have we chosen? VG and PR, okay? So we're going to select all of the cases where VG and PR don't line up with the parental combination. So you can see, again, the parental combinations for VG and PR are that they're both wild type or they're both mutants, okay? If one is wild type and one is mutant, that's not a parental type, that's a recombinant type, okay? So we're going to look for those recombinant types in the offspring. So we can see here at 252, VG is wild type, but PR is not wild type. So that's a recombinant type. And we keep looking and you can find the rest there. Using our formula that we just discussed for RF, and thus finding the map units, right? 1% equals 1 map unit, we can find that it's 12.3 map units separating those 2 loci, those 2 genes. Okay, so this is the diagram that we would be constructed if we used the same methods, right? That same formula. It's good, but it's not great. We still have a discrepancy, right? We have 18.7 here if we sum up consecutive genes, but if we just take a look at the distance between VG and B, just do one calculation for that, we get a discrepancy of 1 map unit. So what's going on there? And this gets back to the double crossovers that I was talking about earlier, okay? Double crossovers, basically what they function to do is make it look like a certain offspring is a parental type, but it actually had 2 recombination events. One to set it off to be like a recombinant type, and then back again to look like a parental type. So what has occurred is 2 recombination events, but it's impossible to detect if you're not looking for it, right? So these are all of the ways that double crossovers can occur, okay? But you can see that there are 4 cases out of these. Don't pay too much attention to all of this. There are 4 cases where it can look like, relative to VG and B, I think we're talking about here, it looks like they're parental combinations, but they're not. They're a double crossover, okay? So now what we're going to do is we're going to do our analysis. Again, we're going to try to map this again, but we're going to try to take double crossovers into account, and we're going to try to come up with a better number. All right? So the first thing that we need to do, you can just see on the far left over there, is determine which gene is in the middle. Double crossovers can't happen between 2 genes that are right next to each other, right? It wouldn't matter like if I made 2 cuts between 2 genes that are right next to each other. It's only when you have a gene in between 2. So I guess what I'm trying to say is if I have 3 genes that I'm analyzing, okay, it's only the outside ones that I need to take into account for the double crossovers. All the other ones we can use our normal method, right? It's only those outside ones that we have to say, where are the double crossovers? Okay, so we need to figure out which one's in the middle and which one's are on the outside. So what we do for that, there's I think a few different methods for this, but what we do is we simply look for the offspring that happen at the lowest frequency, okay? Double crossovers are really rare, and so we're gonna look for the ones that are really rare, right? So we see that VG, B, and then PR plus, so the wild type, is a double crossover, right? And the other one there is too. And so all we need to do is compare those to the parental types. And we can see that the one that doesn't line up with the parental type is PR. PR is the one that doesn't match the parental types. And again, you can look for the parental types right there. All wild type or all mutant. The one that's missing, right, is PR. Okay, so we find that PR is the one in the middle. Okay, great. So now what we need to do is calculate the distance between VG and B, because those are the ones on the outside. Well, we need to do this accounting for double crossovers. All right, so what we need to do is select for all the cases of recombination, right? So it's fairly obvious for 252, 241, 131, and 118. So that's those four at the top of those 6 arrows, right? Like they don't match the parental types very obviously, right? But we can see the bottom 2, they look like parental types, right? They're both wild type or both mutant, again, talking about VG and B, but they're double crossovers. So if we look at the equation for recombination frequency, what we do is we count the double crossovers twice, each of them. Why would we count them twice? Well, there's 2 recombination events happening, right? We have one cut that moves it off, and then another cut that moves the chromosome like back, so to speak. Again, I'd encourage you to look at some diagrams to really help you visualize this. But we need to count it twice. So that's why we get this equation. The top one is incomplete. We need to count the double crossovers. Finally, we would get an 18.7, which is exactly the amount of map units that we would calculate if we added the 2 distances together. Maybe I can go back to that diagram. Yeah, right there. So the 18.7 is what we got by adding the 12.3 and 6.4. So now it matches, right? Now we've calculated this all properly. Okay, so now a question that I want to ask you guys. This still has to do with map units, but it's something that was found in the textbook. I really just want to test your memory, test your knowledge. See if you can explain to your neighbor what chromosomal interference is. Go ahead, I'll give you 30 seconds. All right, let's bring it back. Let's bring it back. Do we have any thoughts? Does someone know what chromosomal interference is? You can scour your notes. Any thoughts? Anyone want to volunteer anything? Okay, this is fairly obscure, so you know what? We'll just go to the answer, okay? So chromosomal interference is the decrease in likelihood of a second crossover occurring in an area where a crossover has already occurred. So if you imagine a synaptonemal complex, right, where we have two duplicated chromosomes that are together, right, they're homologous chromosomes. If a crossover has already occurred between two arms of two non-homologous or two non-sister chromatids, right, if a crossover in a certain area has already occurred, it's less likely that a crossover happens again in the same area, okay? I liken this to rolling a dice. Whenever you roll a dice, there's always a 1/6 chance that you get a certain face, right? The same doesn't apply to crossing over. If crossing over has already occurred in one area, the probability of it happening again is decreased from what chance would dictate. So in the dice analogy, say if I rolled a 5, all right? The next time I rolled the dice, the probability of me getting a 5 would be less than 1/6. That's the way that I put it, okay? So it becomes less likely to get another crossover. And there's a bunch of calculations that you can do with this. I don't want to say that we will or won't be tested on them. It just is in the textbook, so that's why I'm including it. But that's the idea of chromosomal interference. Here's another question that if you were reading your textbook carefully, you may have an answer to. What is the maximum distance in map units that can separate two loci or two genes? Oh man, we got an answer right away. Yes. 50. Boom. That's what I'm talking about. I got the answer on top of your head. Do you care to explain?
I think it has something to do with calculating the average, and then all the time with the average will always add up to maximum efficiency.
Yeah. You're very well versed in your reading. This is exactly right. So the idea is that, say we assumed that crossing over happens like 100% of the time. Like there is never a case where there's not crossing over. Let's look at the different types of crossing over. You can have a single crossover between two given genes. You can have a double crossover between two given genes. You can even have a triple crossover, okay? For each of these different classes of crossing over, if you average them out, okay, and there's different ways that this can happen, and there's a bunch of diagrams in the textbook. Frankly, it's a little hard for me to understand, but they're there, okay? 50% of the time, there will be recombination if you average them out for each of these classes. 50% of the time, will a double cross result in a recombinant type, okay? That's the idea, between two genes. So basically, if you work out these calculations, you can realize that, yeah, the maximum recombination frequency is 50%. Thus, the greatest distance between two genes or two loci is 50 map units. Okay, again, kind of obscure, so I'd recommend reading your textbook for this. I'm just trying to put it out there so it's something on your mind. Okay, so here's the question now. We've been looking at offspring and the numbers of offspring and have been inferring that when there's a preponderance of the parental types, that linkage is happening. But here's a question. How do we know if linkage is really happening? Like, what if it's just by chance that we got this big discrepancy between the parental types and the recombinant types? Like, how do we know for sure that there's really linkage going on? Well, we have a statistical test for that, and it's called the chi-squared test. Now, one thing I want to mention here about the dice, the more times that you do a trial or you do something, so for example, in our case, the more offspring you have, the more that an anomaly is expected, okay? So if I roll a dice 10 times and I get 4 out of 10 being a certain side, okay, say 5, right? That's not too crazy, okay? It's like not what we expected, but you know what? It can happen by chance, right? Like, you know what? We can expect that, right? But if we rolled a dice 1,000 times, right? And 400 of those times we rolled a 5, there would be something up with that dice. That dice would not be a fair dice, okay? So that's something to keep in mind here as well. The larger the sample, the more sure we can be about an effect, okay? So the chi-square test, we're figuring out if an effect is just due to chance or if there really is something going on there. Now, a key term to be aware of is the null hypothesis. Okay, the null hypothesis is that the data that we see is not statistically different between the expected model, that is the predicted values, and the data that we see. So basically, like, there's not an appreciable difference between what we expected if there was nothing that was going on and what we actually got. Okay? Now, if we move on here, we'll also have the alternative hypothesis, but I want to contextualize this within linkage. So when we're looking at linkage, the null hypothesis is that the genes are not linked. Okay, if there's nothing going on, that means there's no linkage. All right. The alternative hypothesis, that is the hypothesis that, you know, maybe there's something going on here that's not just random chance. Okay, that is that the genes are linked. Okay, so how do we actually run a chi-square test? What's the equation that we're using, and what does that look like? Okay, so you can see the chi-squared formula right here. That's the number of observed for a given group or category. I'll make that more clear as we move on. The number observed minus the number expected squared divided by the number expected. Okay, and then you sum that for each class or type. Okay, so I want to put this within the context of a two-point cross. Okay, this is what we were analyzing back at the beginning. Remember, there was just 4 types of offspring, 2 parental types and 2 recombinant types. Okay, if we adopted the Mendelian interpretation of all this, we would expect a 1:1:1:1 ratio between parental and recombinant types. Okay, if there was no linkage happening. That's what we would expect. You can think about why, but it's fairly intuitive, okay? So if we have a total amount of offspring, that's 96 right there, using our 1:1:1:1 ratio, we would expect the offspring to all have 24 in each of those categories, okay? Now, we also put the observed beside that, right? So we can see that we observe more parental types than we do recombinant types. So what we can do is we can plug these in to the chi-square test, okay? And you can just see the number of observed. Let's look at that top row there. The observed, 36, subtract the expected, 24, square that, divide it by the number expected, and then just do that 4 times. And if you run that calculation, you're gonna get all of these. If you sum them together, you're gonna get that number, and that's 12.3, okay? So the chi-squared value for this experiment here on the right is 12.3. Okay, but now we've got to interpret that. Like, what does 12.3 mean? Like, great, 12.3. We need to use a table, and we need to understand 2 more concepts. The concept of the p-value and the degrees of freedom, okay? The degrees of freedom is this idea of how many free choices do you have within the experiment, okay? Let me make this a little bit broad, okay? If I have 4 options, okay, 1, 2, 3, 4. If something is not option 1, not option 2, or not option 3, what must it be? Option 4, right? There was no freedom in that choice. There was no freedom in that. It had to be option 4 if it wasn't 1, 2, or 3. Do you get what I'm saying here? If I have 4 different categories or classes, there are 3 degrees of freedom. And it applies to this rule, n minus 1. For any number of classes n that you have, n minus 1 is the degrees of freedom that you have. Hopefully that makes sense. Okay? So in this case, we have 4 classes, so we have 3 degrees of freedom. We can now look at our table here to try to figure out the p-value. Now, if you have a career in science, which hopefully many of us will, considering that we're science majors, we're probably going to encounter the idea of p-values quite a lot, okay? This happens in psychology. I'm a psychology major myself, so I'll be seeing a lot of p-values too. So it's not just gene linkage where this applies. P-values are very, very ubiquitous, shall we say. Okay, so what we want to do is we want to find the degrees of freedom. We found it was 3, so we look in this table and we find the 3 row. Okay, so we found the 3 row. And then what we want to see is find the range of chi-squared values that this falls into. So 12.3, we can see that that falls between 11.35 and 16.27, okay? And then we just look up to the top and we find the p-value, okay? And we find the p-value is less than 0.01. Okay, what does that mean? Well, the cutoff for statistical significance is having a p-value that's less than 0.05. That's the line that was arbitrarily drawn by scientists a long time ago. They said if our results are not due to chance 95% of the time, it's probably the case that what we're observing is not due to chance, just in general, okay? In this case though, because our p-value is less than 0.01, greater than 99% of the time, it's the case that this is not due to chance. So we can say, you know what, there's something up here. There's something that we can't reliably say is just due to chance. We need to change our hypothesis. We need to reject the null hypothesis. We need to accept the alternative hypothesis. In this case, we need to accept that the genes are in fact linked. All right, hopefully that makes sense. I'm seeing that it's 7:24 here. I usually give a break in between, and we are about halfway through the slides. So I'm gonna say like a 5-ish minute break. Feel free to get some water or something like that, and then we'll be right back. We'll talk about DNA and DNA replication. All right. As stated on the announcement on Instagram, we definitely want to be done in an hour from now. So that's the max length this is going to go. All right. Okay, guys, so while we're on our break here, I want to present to you this idea that I was talking about at the beginning. All right? Oh, man. Okay, this slide right here. Okay. All right. So basically, there's a way that you can proxy your vote. All right, that is give your vote to this group of science students that's going to attend the general meeting. All right, and there's a form that WUSA has sent out. There it is. Okay, and it's at that QR code right there. Okay, so what we would do is you'd fill out the form, and you have to proxy your vote to a specific person. Okay, you can't give it to a group, but what you can do is say down in the instructions section, vote according to the decision of the science students group. Okay, so basically what we're doing is we're taking the democratic power of this many people, distilling it down into a smaller group that will actually attend, but that has the power of the greater group. That's basically what we're trying to do here. So if you're interested in that, again, I'll show this at the end of the lecture, but you can scan that QR code. What you would need to do is put in your name, your email, and your student ID, okay? And then you need to check mark the box that says, I want to proxy my vote to a specific member down there, so just hit yes. And then what you would do from there is you'd put in my information, which is right there, and then in the instruction box, just say, vote according to the decision of the science students group. Okay? So that would be the process if you're interested. All right, we may as well get started with the rest of the lecture here. If you want to keep scanning, you can. Again, I'll put this up at the end as well. I think it would be very cool if just a group of science students showed up at a WUSA meeting with like an insane amount of votes and then just outvoted everyone else. It'd be kind of funny. But it'd be also awesome too, because we could get policies passed that are good for science students. All right, so let's talk about DNA. Let's get going on that discussion. Okay, so briefly we'll just go over the basic composition of DNA. You guys should remember this from high school or BIOL239, whatever it is. But we have nucleotides. These are the monomers of the polymer that is DNA. You can have 4 different types of nucleotides, and they're distinguished by the nitrogenous base that they have on them. There are 4 different types of nitrogenous bases: cytosine, guanine, adenine, and tyrosine, I believe it is. Okay, don't quote me on that, but those are the 4, all right? And now you can have 2 different types of bases as well. There's another distinction: purines, which have 2 rings, and pyrimidines, which have 1 ring. Okay. Other than that, you link these nucleotides together. They're A's, T's, G's, or C's. Again, for DNA, for RNA, there's a U instead of a T. You link them together by phosphodiester bonds. Okay. And it's called the phosphodiester bonds because there's 2 ester bonds and a phosphate in the middle. And you're linking them between the 5' and 3' carbons of the pentose deoxyribose sugar. That's there. You can see the pentagon. Okay. Now, DNA itself is 2 strands that run anti-parallel. Okay. And there are hydrogen bonds between the nitrogenous bases. Between the G's and C's, you have 3 hydrogen bonds, and between the A's and T's, you have 2 hydrogen bonds. All right, so that's our basic information. Now, we're going to take a look at how scientists discovered various properties about DNA, specifically regarding its inheritance. So I want you to pretend like you're a scientist from the past, okay? That's what I'm asking you to do here. Okay, so first things first, scientists wanted to figure out, well, what are chromosomes actually made of? Like, they found them under the microscope. They're like, wow, these things are cool. Wow, these things probably map on to Mendel's idea of units of inheritance because they're conserved generation to generation and they're equally distributed, right? So these are probably our units of inheritance, but what are they actually made of? So what they did is they made a bunch of reagent stains, okay? So these would be stains that color a certain type of macromolecule, but not others. So you could have an RNA stain, you could have a protein stain, but you can have a DNA stain. So they did a DNA stain, and what's left is that the chromosomes are stained bright pink. Okay, so you can see the chromosomes contain DNA. Wow. All right. But now we need to figure out, if we know that chromosomes transmit the genetic information, if they're the heritable units, right? What part of the chromosomes what substance in the chromosomes is what's actually transmitting the heritable information. We know that DNA is in chromosomes, but we also know that there are other materials there too, for example, proteins. So which of those is it that's actually being inherited, and which is just, along for the ride, so to speak? Well, there was a very influential experiment on this, and two of the main experimenters, kind of the preliminary guy was Griffith, and then Avery and some colleagues kept this going, okay? So it's a very interesting phenomenon, and it's called transformation, okay? And there are two bacteria colonies that we talk about when we talk about transformation, okay? We have the S bacteria colony, okay, that stands for smooth, and physically, like if you look at that colony, it will be smooth, all right? And it's virulent, which means when you put it into an organism, the organism gets infected and later dies. Then you have the rough colonies, which are mutants of the S colonies. So if you leave S colonies around for long enough, they get a mutation which leads to rough colonies. And physically, if you look at them, they're rough appearing on the surface. Okay, so the experiment here, we're using these S and R colonies to figure out the nature of inheritance. So you can see we have three controls on the top here. The first control is when you inject the S colony into an organism, and obviously it dies. This is what I just said previously. Okay, we're looking at a mouse here, or a rat, one of the two. If you inject an organism, a mouse, with the R colony, it lives. So there's something about the R colony that's not virulent. It doesn't infect that organism. Okay. Then, if we look at the third control, if we heat kill the S colony, the virulent colony, if we heat kill it and inject that into an organism, the organism doesn't die. There's something about heat killing it, breaking apart those bacteria that makes it non-virulent, okay? So that's another control. But here's the kicker, okay? Here's like the, oh man, what on earth is going on experiment. We take heat-killed S bacteria, inject them into a rat, but then also inject that rat with R bacteria, okay? So in both of the controls, we found that this didn't harm the mouse or the rat, right? But when we inject both together, it does kill the rat or the mouse, okay? What on earth is going on there, right? Our controls tell us that this shouldn't be happening, but somehow it is. Well, Griffith, who ran this experiment, took a look at that dead mouse, okay? And he found living S cells. Boom, like what on earth is going on? Because I only put in, R bacteria and killed not alive S bacteria. So what's happening? That we have live S bacteria. Well, Avery decided to look into this a little bit. He's like, man, we're gonna figure out what's going on. We're gonna find what's called the transforming principle. Because it's transformation that's happening here. We have some heritable information moving from those S bacteria into the dead S bacteria, into the live R bacteria, and changing them back into S bacteria. So there's some exchange of heritable information going on. And really the question is, what material is it that actually allows that transformation to occur? So through a bunch of experiments, and this took quite a few years, I think it took 15 years for this team to figure out what was going on. Avery, McLeod, and McCarthy, I remember those from high school, they were able to purify the transforming principle. They were able to purify the macromolecule that was responsible for this transforming, okay? But they needed to figure out what it actually was. Like they had it in a tube, but what is that, right? Well, they were able to do a physical and chemical analysis, and they thought, this looks a lot like DNA, right? But if we keep our hands and feet within the experiment, how experimentally can we show that this is indeed DNA? Well, what we can do is we can take that transforming principle and subject it to enzymes, okay? Now, there are enzymes that break down various types of macromolecules. You can break down proteins, you can break down RNA, DNA, or lipids. So they added these various ases to the transforming principle, and they tested them for transformation. Can transformation occur if we take out this type of macromolecule? Well, transformation could occur in all cases except for when DNA was destroyed. So they said DNA must be the transforming principle. That must be the thing that's causing this transforming to occur. Unfortunately, this did not convince everyone in the scientific community. So there was another set of experiments that were undertaken to try to show that, in fact, DNA is what holds the hereditary information. And so here we look at viruses, okay? And we look at the T2 phage specifically. You can see the structure here on the left. It reminds me of a spider, right? Like, you know, another image is a syringe, okay? Why a syringe? Because you can see the capsule at the top contains DNA, and the outer structure is made of protein. And the DNA is shot into the host cell, okay? So that's, in that way, it's a syringe. That's the physiology of the virus. Now, something that was discussed in class, and it's a very interesting question, is a virus like a living thing or not, right? And I guess the conclusion is that it's not, but like kind of, because it can reproduce, but it can only reproduce with the help of a host organism. So it's like a macromolecule just sitting there unless it has a host organism, and in that case, it does very much function like it's alive, okay? So let's look at the life cycle of a virus here. We have a host cell at the top, and a virus attaches to that host cell and injects its DNA. Its DNA is then incorporated into the DNA of that host cell, and when transcription and translation occurs, the genes that are encoded in that viral DNA are expressed. And what do those genes code for? They code for the components of the virus. So they just code for making more viruses. And you can see that in the picture here. There are virus parts and they're becoming assembled. So now we just get the unstoppable production of viruses. And they keep building up within the host cell until the host cell ruptures. It bursts. It can't hold them anymore. So this ultimately kills the host cell. But what it also does is it allows the spreading of these viruses to new host cells. It multiplies the hosts, the viruses. And so if left unchecked, i.e. if there's no immune system, then this virus cycle can just continue uninterrupted and kill any host organism that it's in. Okay, so let's look at the experiment that uses those mechanics and those characteristics of viruses to prove that DNA is what carries the hereditary information, okay? So we have our T2 phages here on the left. Now the strategy is to distinguish between protein and DNA. And how do we do that? We label the protein and the DNA. And we do that radioactively. So what you do is you allow you allow viruses to propagate inside a medium that contains radioactively marked phosphorus, okay? And the phosphorus will be incorporated into the DNA. So it will mark all of the DNA radioactively. It won't be incorporated into the proteins, just the DNA, okay? On the bottom, you can see that instead of growing these viruses in phosphorus, you grow them in radioactive sulfur. And that sulfur is incorporated into the proteins, not the DNA, just the proteins. So you can see that what results from this process is radioactively marked DNA in the top T2 phage and radioactively marked protein in the bottom T2 phage. Then what we do is we simply infect a host cell and then examine the host cell after that infection. So if we look at the top there, the DNA has been inserted, and then if you examine the cell, you'll find the radioactivity in that host cell. And since the radioactivity was put into the DNA, we know it's the DNA that's inside that cell. Conversely though, if we look at this host cell and we try to find the radioactivity, we won't find the radioactivity inside the host cell. We'll find it in the ghosts, they're called. That is, the phages that don't contain DNA anymore, but are still just existing in the environment. That's where we'll find the radioactivity. So through labeling with radioactivity, we're able to find the macromolecule that is inserted into the host cell, and we're able to determine that that is in fact DNA. And since whatever substance that is put into this host cell causes the propagation of the virus, that is, the making of new viruses, that's kind of the definition of hereditary material, right? That's what we should be looking for if we're looking for the thing that causes inheritance. So this was really, really good evidence that DNA is what causes inheritance and the propagation of viruses. One last thing to take a look at, you'll be very familiar with these researchers here on the right if you've ever looked at genetics before. Crick, Watson, Franklin, and then Wilkins, who's kind of the boss in this picture but didn't do much of the research, is what I've been told. So what we're looking at here is the actual structure of DNA. We talked about this on the first slide in the DNA section. DNA is a double helix, right? It has certain bases in it. There's a given structure, a prescribed structure for DNA. So how did they discover what this structure was? Well, Rosalind Franklin here was able to take an X-ray diffraction picture of DNA, kind of from the top, right? If you look down on the strand of DNA, this is what you would find with an X-ray diffraction picture. Now, she wasn't able to interpret the image and really didn't get much credit for this, which is unfortunate. But Watson and Crick were able to interpret this X-ray image. They had the knowledge to say, man, that looks like a double helix structure. So what we should be interpreting this as. And so with that expertise, they were also able to figure out that there should be equal amounts of A's and T's and G's and C's in all DNA samples. So they figured out that these bases are what pair together. And of course, that there's this double helix structure of DNA. So very significant contributions. The final slide here in our DNA structure section is that there's not just one type of DNA. There are actually multiple types of DNA. The other ones are just very rare. So the B form DNA is what we all recognize as DNA, okay? But we also have the A form and the Z form. I don't really want to read too much into this because I'm not an expert in this at all, but I can see that the A form is a little bit more jumbled in terms of its connections. And the Z form, this is actually different. It's left-handed. So I want to explain handedness to you, okay? If you take your right hand and curl it like this, the rotation, the direction of rotation is counterclockwise, okay? My thumb is also pointing upwards. So as the DNA moves in a counterclockwise direction, it moves upwards. You can try that with either of the images on the left, the A form or the B form. If you do this, you'll see it curls upwards, okay? But if you look at the Z form, it actually is not right-handed, it's left-handed. So as you rotate in the clockwise direction, you move upwards on the DNA strand. So fascinating, but something to take a look at. I see some people with hands, that's good. All right, now we move into our final section. We're actually very well paced today, which is good considering that this is some tough material. So we should be done rather soon. That's encouraging. Okay, so the first question I have for you, which is a very simple one, you shouldn't really need to do this on paper. If a strand of DNA is 35% adenine, what is its percent composition of guanine? Go ahead. Has anyone come to an answer? Just put your hand up. Yes. 15%. Let's see why. So we know, we talked about adenine, right? Yes, adenine. We know adenine pairs with thymine, right? So we got to multiply 35% by 2. The A's and T's pair, okay? So the G's and C's must be 30% of the bases. And if we divide that by 2, because for every G there's a C, and for every C there's a G, we get 15%. Does that logic make sense? The fact that you use is that G's and C's always pair together, and A's and T's always pair together. If you logic with that, you can very easily come up with this answer. I'm not the TA, but I would not be surprised if a question like this just showed up. It's a very, very nice multiple choice question. Simple to calculate, but if you don't know what's going on, you're lost. Okay, so now we come to the significant question of this, which is how does DNA replicate? By what model do we get from one strand of DNA to two? How do we prepare for meiosis or mitosis in S phase, right? What are we doing to make two chromosomes out of one, or double a chromosome? All right, well, the main experiment in this with this subject is the Mendelssohn-Stahl experiment, which concluded that DNA replication occurs in a semi-conservative manner. And I'll define that for you very shortly. But I want to start with the experiment here. So these are the conditions that we use to create the experiment. You take bacteria and you grow them in a medium of heavy nitrogen. So that is nitrogen that has one extra proton, okay? You grow it in there for quite a while so that that heavy nitrogen is incorporated thoroughly into the DNA. So now all that DNA contains heavy nitrogen and not any light nitrogen, okay? Then what you do is you transfer all that DNA that's marked with heavy nitrogen into the light nitrogen medium, so that's in red there. And you see what happens. You look at the weights, the masses of DNA that are produced. So let's take a look at this here. This is the model of semi-conservative replication, right along the top there. At the beginning, you have two strands that are made of heavy nitrogen, right? If you were to assess the mass of this, so what you would do is you would break apart those bacteria, isolate the DNA, and then centrifuge that DNA for like a really long time at a really fast speed. What you can do is get these readings on the bottom here. The further right you move on this spindle thing, the more heavy the sample is, okay? So you can see from this analysis on the far left, the heavy just has one band, right? So that's what we would expect if we have DNA that's just been grown in that heavy medium. Now, if we let one generation of replication occur and we analyze the DNA again, the semi-conservative model would suggest that both strands should be composed of one strand that's heavy and one strand that's light, okay? That's what the semi-conservative model would say. And what we would expect from our mass measurements, from the centrifugation, is one band that's half heavy and half light, that kind of mass. And we see that right there. It's to the left of the heavy line. Now, if we do a second generation, so if we're thinking like alleles here, right, we would say like the F2. I don't think you can really apply that in this situation, but you can see here that the semi-conservative model would suggest that we should have two DNA strands that are both light, and then we should have two strands that are, again, hybrids. And that's exactly what we see from our mass analysis. We see one band of light nitrogen and one band of hybrid between the two, okay? So this is really good evidence that the semi-conservative model is what indeed happens for DNA replication. The semi-conservative model. Hopefully you remember that. Okay. So I just want to show you the other models that were considered. There's the semi-conservative model, but there's also the conservative model, which is just that one of that first generation strands of DNA is heavy and one is light. That's it. Now, the bands that we would expect from this, right, would not be a hybrid in the middle, but would be two bands. One that's this band and one that's the one on the far right there. Right, we'd expect two of those, not a hybrid one. So that's what's evidence against that one. And then another theory that's rather jumbled and would produce some very interesting weight measurements is this dispersive one, where you have these chunks of DNA all spread throughout. Maybe the hypothesis is that they're actually all the same weight, which was also not observed. So we can conclude semi-conservative replication. Okay. So now let's look at the actual mechanism here. What components are involved in truly replicating DNA? So I've just assembled this quickly this afternoon. So this is just a very cursory overview. And frankly, what I suggest that you do is you find a whiteboard or also just a piece of paper and try to draw out these diagrams. Try to draw out DNA replication occurring. Draw in the components that are involved and the order in which they work. I will go through these quickly here, but I encourage you to do this on your own. So the first step of DNA replication, there are two of them, is initiation. So initiation occurs at the origin of replication, and the origin of replication is an area that's rich in A's and T's, okay? Not G's and C's, for some reason. There's probably an explanation for it, I don't know it. So at this origin of replication, an initiator protein will bind there. We'll say this is our origin of replication. From there, DNA helicase is recruited to that origin of replication. And what it does is it cuts the DNA apart. It unzips it, so to speak. And it creates what's called a replication bubble. Okay, you can see that bubble there. At either end of the bubble is a replication fork. And you can understand why it's a fork, because two of the strands meet. Okay? All right, from there, single-stranded binding proteins are added on each fork, and they're used to keep the forks separate. So they're used to keep the DNA strands from zipping together again. That's what they're there for. And then finally, in order to prepare for elongation, the second phase, RNA primers are added at the site of the origin of replication, okay? And the reason that we need primers is that the molecules which are going to add bases, complementary bases, to each of these template strands, it needs a place to start. It needs some base to continue adding on to. And so RNA primers are that primer, or that starting place. And the molecule that's going to add primers is primase, okay? Wow, it's pretty easy to remember, so don't stress yourself about that. All right, so once we've added those primers in, now we begin elongation, okay? And elongation is the addition of complementary bases to the template strands that we have, each of those strands of that original DNA molecule, okay? Synthesis always occurs in the 5' to 3' direction, always, okay? And this will be done by DNA polymerase 3 initially. It will find that RNA primer and then synthesize in the 5' to 3' direction. Now, what you'll notice is that because DNA is anti-parallel, we run into a problem. We end up having what's called a leading strand and a lagging strand. So the leading strand is very simple. You add in an RNA primer, and the 5' to 3' direction of synthesis happens to correspond exactly to the direction that points towards the replication fork, okay? So synthesis occurs just like very smoothly in that direction, towards the fork. But on the other side, on the other strands, you can see that's on the bottom this time. The 5' to 3' direction of synthesis points away from the replication fork. It points in the direction of the other replication fork. You'll notice in this diagram we only have one fork, but if you went on the other side of this, the other side of the bubble, you would find another replication fork. Now, how do we resolve this, right? Like, what do we do when the synthesis of the direction of synthesis is the opposite way from the fork where we're expanding? Well, what we do is we have Okazaki fragments, okay? So we continually add in new primers as the replication bubble expands. So as it expands, you add in a new primer, closer and closer to where that replication fork is. And then the DNA polymerase 3 is able to just synthesize in that direction away from the fork, okay? Now, what we need to do also is get rid of all these primers, right? If we had all these primers littered throughout our copies, that would be messed up because they're RNA, they're not DNA, right? We're not making a proper copy. So what needs to occur is the action of DNA polymerase 1. DNA polymerase 1 is able to find the RNA primers, remove the RNA primers, and then replace them with DNA bases, not RNA nucleotides, okay? Now, the thing is, why does this work, right? Why do we not need primers again, right? Well, it's because there are surrounding DNA sequences, right? It's like we already have the starting spot there. We're not starting with an open expanse. And so it's really easy to just fill that in, okay? Now, the final thing we need to do is connect all of these fragments, right? All these Okazaki fragments, they're called. Named after the experimenters. They were a husband-wife duo from Japan, very interesting. And so we just need to connect them all. So there's a protein called ligase that does that, and it just connects the sugar phosphate backbones to create a singular piece of DNA, a singular strand. Now, the final question I have for all of you today is, what is telomerase? And maybe another question that kind of accompanies this, what's a telomere? That's the question you're going to have to answer first. So talk amongst yourselves. What's telomere? What's telomerase? Try to explain the action of telomerase. You can see this gene. Remember earlier? All right. Do we have any explanations here? Is someone willing to volunteer an explanation? What did you guys talk about? There's a lot of talking going on. I hope it was about telomerase. Yes.
So, telomerase catalyzes the synthesization of telomeres, which exist at the end of either chromosomes, like DNA fragments, and it's like a long chain of bases that don't proliferate.
That's right. They're protective ends. So the telomeres are the protective ends of every chromosome. And the telomerase is what supports the maintenance of those telomeres. So here's the fundamental problem with the telomere. Like you said, it's a repeated sequence of bases, okay? The sequence GGGTTA, okay? And that repeats like a bunch of times. Like we're talking over 100 times here. And it's kind of like a buffer. Why do we need a buffer at the end of a chromosome? Well, if we think about the process of DNA synthesis, right? We're removing the primers, right? And we're replacing them with DNA. But if we look at the ends of the DNA that we've synthesized, if we remove the primers on the ends, there is no way to synthesize replacements. There's no way that we can move in the 5' to 3' direction because we have nothing to start with. We would need another primer. Do you get what I'm saying here? Like there's no place to start the 5' to 3' addition of new bases. So what you get after just one generation of DNA replication are the loss, the gaps at the ends of each DNA strand. You can see that there, right? They're just missing this sequence of bases. And it would be really, really sad if we started losing our DNA, because man, our genes would stop working, we would stop working, you know what I'm saying? So we have telomeres. Now, if you had telomeres and you perpetually degraded like this, eventually you would start compromising the chromosome. And this is actually the case for various somatic cells in our body, that they only last for a certain amount of time, and this is what determines their best before date. But for some chromosomes, it's really important that they stick around, like forever, i.e., the chromosomes in our gametes, right? In our sex chromosomes, right? Those are important. And so we need to have some way of preserving the telomeres in certain chromosomes. And so telomerase is what's able to do that. Now, what I want you to see here is just this image on the right, okay? You can see an overhang right? The overhang that results from removing the RNA primer at the end, but not being able to replace it because we have no way of starting 5' to 3' synthesis. What telomerase does is it allows the increasing of that overhang, like it increases the size of the overhang of bases. So what telomerase is made of, it's made of a protein, but also a little piece of RNA. That RNA is actually exactly complementary to this repeated sequence GGG-TTA, okay? And so what it does is it allows for this extension. So you can see that the overhang is extended here, all right? Now, when we've created enough of an overhang, what we're able to use is the RNA from the telomerase as a primer to synthesize back 5' to 3'. Do you get what's going on here? We're extending the length of the overhang such that we've gone past the end of it, right? We have some space here. Then we're just synthesizing back all this green section here. And then if you wanted to, you could just cut that off, right? And now you have a fully preserved chromosome. Pretty cool, eh? Okay, that's all I've got for you guys. Thanks for coming. All the best in your studying. I really hope it goes well. And thanks, guys. Thanks. It was a tough lecture this time around, so really, thank you. And finally, it would be really cool if you guys got in on this. And if you want to be part of this group of science students that does go to the WUSA general meeting and decides what to do with this large vote, please just come up and talk to me. We'll get together, we'll read through the agenda, we'll figure out what to do. If you're not planning on coming, please consider just proxying your vote. I'm going to put that on my Instagram as well. All you have to do is fill out that form, okay? Thanks, guys.
I had to leave because if not, but
Yeah.
so you covered linkage.
Yep.
You covered DNA mapping.
Yep.
I think that's as far as I was with you with before I could go.
Okay, so what I want to do here,
Yeah.
I've been recording this.
Oh really?
Are you gonna post it?
So I want to post it. That's right. I recorded my last lecture. Let me just go here and stop it.